Hello, this is Dylan Tang, and today we're going to be talking about mitochondrial diseases and how they relate to CRISPR. So, let's go over the basics of mitochondria. As we know, mitochondria in our bodies or our body's cells have their own DNA. Not all the mitochondria in our body have the same DNA. So we could have, for example, let's say we have a cell and it has 10 mitochondria. Each individual mitochondria in that specific cell have their own DNA. And they are not identical to each other. What we also know is that the mitochondria are inherited from our mothers. Only from our mothers, not the father. Only the mother. What does this mean? Well, this means that in a single cell, for example, there are multiple mitochondria with their own DNA. These mitochondria can have genetic mutations that can affect the specific mitochondria with that DNA from functioning properly. So let's say in a cell, as I said before, 1 out of the 10 mitochondria have a mutation, and this mutation prevents it from functioning properly. So in that cell, only 9 out of the 10 mitochondria function properly. What does this mean? Well, if the mitochondria doesn't function properly in a cell, then the cell doesn't produce as much ATP as it should. So, usually the disease won't affect every single mitochondria, except those that have the genetic mutation in the DNA. So the energy produced will be decreased, causing problems in bodily function, since all of life's processes require the use of energy, which comes from the mitochondria. Since mitochondria, ATP, and the body uses ATP. Well, organisms use ATP. With this information, we can see how the mitochondria found in our cells come from our mothers. This means if the mitochondria with the genetic mutation is present in the mother, it will be present in the rest of her offspring. Sometimes the mitochondrial mutations can occur randomly, although with a smaller chance of happening, these mutations can occur. So, even if you don't have it, there's still a chance for some mutation to happen and you'll have dysfunctional mitochondria. And so once these mutations show up in your offsprings, there's a chance to also inherit the mutated mitochondria or dysfunctional mitochondria. However, since mothers are the only ones that can inherit it, so let's say if the father has the mitochondrial disease but the mother doesn't, then the offspring shouldn't have the mitochondrial disease. These mutations can affect the mitochondria's ability to function properly, because defects in the genes that impair the ability of the mitochondria to make proteins that use oxygen and produce energy can be affected by both the nuclear and mitochondrial DNA. So the cell's nucleus, the DNA in the cell's nucleus, if that's affected, it can also affect the mitochondria. And the mitochondria itself also has DNA, like its own DNA. So some various forms of mitochondrial diseases include mitochondrial myopathy, myoclonic epilepsy, Leber's hereditary optic neuropathy, and Leigh syndrome. Most symptoms that come with the mitochondrial disease include fatigue, weakness, cognitive disability, and other symptoms. This is because mitochondrial diseases cause the body to produce less energy than a normal human. So if the body is lacking in energy, it will have problems functioning properly. So for a normal human, running and doing all the normal human activities, that'll be really, natural for them. But for someone that has a mitochondrial disease, they're probably going to have a lot more trouble doing that because they're not producing enough energy for the body to use. So organs that have more mitochondria, aka those that require more energy, are more heavily affected by these mitochondrial diseases. So organs such as for example, the heart, kidney, and brain are some of the most heavily affected organs. So people with mitochondrial diseases will show the most symptoms in these organs. Now, let's move on to CRISPR. So CRISPR clustered regularly interspace short palindromic repeats, or CRISPR for short, CRISPR, is a process where it essentially cuts the specific DNA sequence which contains an error or unwanted sequence of DNA, and then it allows the DNA to naturally repair itself. So some general parts of the CRISPR that you need to understand is the Cas9. That is an enzyme related to CRISPR, which is the part of the system that cuts the specific DNA you want out. Next to the Cas9, there's this guide RNA, which basically is a sequence of RNA that can attach to which is a type of RNA that binds to Cas9, and according to the RNA that it binds to, it will show what it will cut out. So there are three ways to use CRISPR. There's disrupt, delete, correct, and insert. For CRISPR, disrupting causes gene inactivation by creating a cut, resulting in addition or deletion of base pairs. The CRISPR will come in here, it'll cut out this section of the DNA, and then after that, it'll add a little sequence of base pairs that causes the gene to be inactivated. So for deletion, CRISPR can also just be used to cut out CRISPR can be used to cut large parts of DNA where it cuts at two different sites, and then it unites the two, and then deletes this specified gene sequence. And then the last way you can use CRISPR is to correct and insert. By adding a DNA template with CRISPR Cas9, it allows the cell to correct the gene. CRISPR goes in and cuts out the section of DNA, and then it inserts a part of a DNA sequence that you want to be coded into it, or fix the DNA sequence that may or may not be mutated. So when it comes to mitochondrial diseases, it can potentially be fixed by allowing CRISPR to go in and edit and repair the mutated genome, affecting the ability of the mitochondria to function properly. So in time, mitochondrial diseases may be curable.